Alright, so today we are going to talk about plant tissue culture application. So there are so many things that can be done with plant tissue culture. So these are the things that we are going to look at: suspension culture, organogenesis, haploid production, short-term, and long-term conservation, somaclonal variation. So for organogenesis, organogenesis is production of new organs which was not present in explant previously. For example, if you take a look at this plant right here, initially it starts with a small cut. Then you put it on the media, then it grows into callus, and eventually in the end it becomes a complete plant. Okay, so that's we say that this part is basically morphogenic or organogenic, and the new organs such as shoots or roots which are induced to form and on cultured plant tissues are known as adventitious shoots or roots. So this one is it's coming out from the explant, so they are known as adventitious shoots or roots. And this is the figure that shows how from a stock plant we can get from any parts, for example the meristems, the explant from various tissues, and they eventually will become a whole new complete plant. Okay, so the plant part is basically called morphogenic. Next one, you move on to somatic embryogenesis. So usually when we have somatic cells, they will turn into a normal plant, but they will not produce embryo. But in this case, if you grow tissue culture, you can actually induce them to become somatic embryo, and somatic embryo is actually called synthetic seed. Okay, it's a seed. It is like a seed because it has an embryo, but actually it is coming from somatic. It doesn't have any representatives from the male or the female, but it's just it formed embryo, so we call it synthetic seeds, and it is coming from somatic cells. So the somatic embryos may arise directly from the explants or indirectly after callusing. And somatic embryos resemble zygotic embryos morphologically. They are bipolar, meaning they have two points. One goes up and the other one goes down, and we're typical embryogenic organs. So the one that goes up is the shoot, and the one that goes down is the root. It is not connected to the explant or callus cells by vascular tissue. And it allows for large-scale vegetative propagation, and usually it can be scaled up by using bioreactors. And somatic embryos can be cryopreserved, meaning it can be stored in cold condition. So this is how it looks like: 2-cell becoming 8-cell, 32, and eventually complete cell. Okay, and another example of organogenic traits or somatic embryos is you take a piece of a mature plant and you culture it on the media, and then it grows bigger and eventually becomes torpedo stage and cotyledonary state, and eventually it becomes a complete full plant. So when it is during its root cell culture, they become embryo, somatic embryos. So in these somatic embryos resemble embryos that we have like it was in the seed. So that's why they say it is the synthetic seed. Meanwhile, for callus and suspension culture, Callus is a growing mass of cells, which it looks like something granular, and it looks whitish. And so when explants are cultured under correct conditions and appropriate solid medium, and they will produce callus. Okay, so this is how callus looks like, and it looks granular. The singular is callus, and the plural is calli. Alright, so how everything takes place is from callus, it differentiates into organs. However, if you, let's say you take a sample of an organ and you culture an organ, it will de-differentiate into callus and become something else. Okay, if let's say you take a leaf, which is an organ, and you culture it, it will de-differentiate, meaning from something reverse to nothing, and it becomes something else or something. Okay, so that is what de-differentiation means. Say it one more time. If let's say you have a root, initially you have a root and you culture it, the root cells will reverse to become nothing, and it will become callus, and the callus will return, it will become something else as it differentiates. So the callus culture could be compact or friable, meaning it could be hard, or it could be something that can spread around. Okay. Compact callus shows densely aggregated cells, while friable callus shows loosely associated cells, and it breaks apart easily. So you have two types. One is compact, really compact, and the other one breaks easily. So embryogenic callus allows for regeneration of plandets through organogenesis or embryogenesis. Okay. So on the left side, which is A, okay, and A is embryogenic callus, which you can see that it forms like an embryo, looks like an embryo, and on the right side is non-embryogenic callus. It doesn't look like embryo. Embryo should look like a seed, like an embryo that sits in the seed. There are three stages in callus culture. Number one is induction. You induce by de-differentiation, meaning, like I said before, and from an organ it returns to become nothing, and it turn out to be something else or something. And then there is proliferative stage, which the cell divided rapidly. Then you have morphogenesis stage, which it differentiates into something. So this is callus proliferation. It's from let's say you start off with a seed, and as it gets bigger, it looks like something granular on the media. And these are regeneration of plantlets from callus. So the whitish, the ones in white are callus, and the one in green are the shoots. Okay, the shoots which has formed from the callus. For callus culture, calli produced from the wounded sites of an explant. If let's say you want to grow, let's say you have tobacco leaf, and then you cut it on the side and you put it on the media, and on the leaf, basically on the side of the leaf, let's say it's a square shape. So on the every side, on every edge of the leaf, callus will grow, because it is basically the wounded side of the explant. The wounded site is used as the target for plant transformation in genetic engineering, because bacteria can only go into something through a wound. And next is the habituation, the loss of the requirement of auxin and cytokinin by the culture during long-term culture, meaning after some time you become immune to the amount of the hormones that you supply, and they have no effect in the end.
And actually, there are so many things that you can do with tissue culture. And for example, if let's say you have a plant initially you do genetic modification, and the next thing that you need to do is tissue culture, because once you have the gene modified, the gene has been modified, the next thing you need to do is to grow the tissue until it gets bigger, because the first thing you need to do is modify the gene, and then you transform the gene into the plant, and then you grow the plant in a tissue culture media, and eventually they will grow bigger, and you can do all sorts of things, such as growing callus, growing suspension cell, or probably if you think that there is an interesting compounds that you want to overproduce, you can grow them in cell culture bioreactor. Okay, and in the end you will produce a lot of products through this technique. For Callus and suspension culture, you can look at this figure right here. Callus is something that Callus is something compact, and suspension culture is something that is friable. It is like a callus that is mashed, and you can find it everywhere in the media. It's in order for you to produce cell suspension culture, something that you need to know is suspension culture is something that is friable. The callus is friable, meaning it is isolated. The cells are isolated, and it is everywhere in the liquid media, and it is grown in a liquid medium, and it is agitated so that it is called suspended. Okay. Suspension culture must be constantly agitated at 30 to 100 RPM, so that it can constantly eat. So that's why when you agitate, the cell is able to eat from the media, because it does not have any limbs. So that's why you agitate the cells. So this is how it looks like. It's friable, and it looks like some cells that has been broken through agitation. A cell suspension culture consists of single cells and smaller group of cells suspended in a liquid medium as a result of the mechanical impact in agitated liquid media. So once you every time you agitate your media, so all of the cells, they become broken into single cell. So suspension culture grew much faster than Callus culture because it is constantly being exposed to the nutrient. And the media usually contains 2,4-D or auxin, okay, and pectinase can be added to the media and improve the production of single cell. Okay, so pectinase is another important chemicals that is usually added in order to ensure that single cell is produced. So this is how it looks like from an organ and tissues, and then you surface sterilize, and you produce Callus culture, and then you chop it, and you get a cell suspension culture, and then you can separate the cell suspension culture through filtering, and then decantation or addition of pectinase, and in 5 days or 2 weeks you will get a fine suspension culture, and after that you can weigh them up, pipette it, pour it, and that's how you get suspension culture. This is the normal growth curve of any living thing usually. So you started off slow, and this is the period where you are building up your DNA, and once everything, the building blocks is ready, it will become increased, the production of there will be an exponential phase of growth. So imagine if you have imagine the situation of the COVID-19. So this is the incubation period. Okay, incubation period, and then this is the growth phase, exponential phase, and this is where the newly made cells is equal to the cells that is dying, and this is the stage where the dying cells are more than the newly made cells. Okay. So applications of suspension culture, you can overproduce a lot of secondary metabolite by using bioreactor. For example, there is something that you are interested in such as, let's say, caffeine, and you want to overproduce the caffeine, so you can do it by putting it in the bioreactor. A bioreactor is a glass or steel vessel. So this is how it looks like, a bioreactor. So you can put the suspension culture in here, and the pH is maintained, the oxygen is also supplied, and everything is controlled, and it is sterile, so that the product or the secondary metabolite is produced at an optimum amount. Next one is protoplast. Plant cells have had their cell walls removed enzymatically by cellulases and pectinases. So this one, sorry. So you can, this is pre-digestion and post-digestion. You can see that the membrane has been taken out. So what's left is just the cytoplasm and the nucleus. Protoplast fusion is, if you still remember, let me show you right here. Protoplast fusion is, sorry, okay, I'm sorry. Okay, this is the one that I've shown you before. We found it from YouTube.
This is like the most ridiculous thing I've ever done in my life.
Protoplast fusion. So you, by looking at the microscope and doing this,
1 minute,
and you are injecting into the nucleus the new genetic information that you want to transfer into.
It just seems it's like always me, you know?
Well, it could be me, and then you say that.
No.
Oh, you take some time after pressing. That's what I have to do.
Okay. So basically, this will produce new hybrid plant with the characteristics of both, because you are fusing two nucleus together, and the hybrids can regenerate a wall, be cultured, and produce a hybrid plantlet. Next one is haploidy. In this case, only it has a single set of chromosome instead of two. Imagine yourself, you have 46, but in this time they they try to make it 23, so that it is easy for them to cut time to do breeding programs. And there are several ways for you to produce haploid production: androgenesis, inactivated or eliminated before fertilization.
Double haploid process for over 10 years. Each year we use it more and more to develop new inbred lines. The traditional inbreeding process takes on average 8 generations or 8 cropping seasons to develop an inbred line that is 99.6% inbred. Comparatively, the double haploid process only takes 3 generations or 3 cropping seasons to develop an inbred line that is 100% inbred. Before we talk about the double haploid process, let me explain 2 terms. Haploid organisms are individuals who only have 1 set of chromosomes. Diploid organisms are those that have 2 sets of chromosomes. We get 1 from our mother and 1 from our father. Humans are that way. corn is the very same in that every individual in a corn field has two sets of chromosomes, one from the pollen parent and one from the ovule parent or the female parent. So the process of doubled haploids or developing inbred lines through this process really involves two steps. The first step is creating haploid individuals. So this is accomplished by pollinating our breeding populations with a special variety of corn that when it pollinates our breeding populations, it creates individuals on those ears, individual seeds that are haploid. They only have half the normal amount of DNA, and it all comes from the mother plant. So the second step involves taking those haploid kernels. We germinate them in a special solution that effectively doubles the number of chromosomes in the cells. And if those cells continue to divide and produce a tassel and an ear shoot, and we can pollinate that plant, the seed that's produced will be an instant inbred line. So in addition to speeding the rate at which we can develop new hybrids, all of these technologies that we deploy today in our breeding programs are much more precise and effective, and ultimately it allows us to deliver higher quality products in terms of purity and performance. The double haploid process is ultimately important to the farmer because it allows us to develop new products, better quality products at a much faster rate. So at the end of the day, our farmer customers, they can just expect better products every single year.
All right, so that's about haploid production. So basically he's trying to say that it started off with being haploid, or half of what they have, and by inducing with a special material, and then when they have it haploid, so it is easy for them to breed with another to hybrid them with something else. Okay, if let's say you have two corn, you have produced a haploid which is half from what it is now, and then you combine them with something else without having to wait until 8th season. So that's haploid. So the in vitro techniques that you have is androgenesis, which is by using the male, and gynogenesis by using the female. Androgenesis is production through the enter or pollen culture. So the pollen can be extracted. You take the pollen, and then the wild pollen is collected, and they are cultured on solid medium, and so the callus or embryo form is transferred onto a suitable medium. So eventually it produces a haploid plant, and they will treat it with a colchicine in order to produce a diploid plant. So if you think about it, how it shouldn't survive in the first place, but because they treat it with colchicine. So if let's say the genetic material is, let's say, big T, or if let's say you will have a homogeneous gene or homogeneous allele. Okay, so parent plant selection of flower bud, and you sterilize a young flower bud, and you can regenerate. So you get it from the male gametophytes, such as the anther. So imagine you have the anther, you grow the anther, and then you add colchicine in order to produce homogeneous diploid after you produce the haploid. So it's either pollen or anther. It's half, and so eventually they put colchicine in, and it will become diploid. So keep past this. Next one is gynogenesis. This one is a bit harder because you are using the ovary. You are actually culturing the ovary, but this one is not preferred unless it is hard for you to do androgenesis. Why is it not preferred? Is because the ovary is really small, and there is only one ovary per one flower, so it makes it quite hard for you to do the process. So most of the time, gynogenesis is less efficient than androgenesis, and it is used in plant families that do not respond to androgenesis. The major limitation of gynogenesis is because, like I said, one flower only have one ovary. The dissection of unfertilized ovaries is rather difficult, and because one ovary, one flower. Next one, we move on to short-term and long-term germplasm conservation. Um, This is to preserve the genetic diversity of specific plant or genetic stock for future use. So when you say germplasm conservation, that means it is trying to keep your samples in somewhere safe for a long time. There are two ways for you to do it. One is in situ. For example, you leave it on its natural habitat, so that you preserve its everything, so that it can live in its own condition, its own environment, such as in the park or in the zoo. Another way that you can do is by ex situ, which is gene bank or seed bank. So for in vitro, the ones we just have we looked at before was ex situ. Sorry, the one we looked at was this one. This was the one that we talked about, and this one is if let's say the one that we talked about was ex vitro, so this one is in vitro. So this one, let's say you want to do cryopreservation, so you can take a look at the first figure here. It shows that it is, you can see vapor, something like something that has smoke in it. Actually, that is liquid nitrogen. It has a temperature of minus 198 degrees Celsius. So everything that goes into liquid nitrogen will be cryopreserved. It will be very, very cold. And you also have cold storage in the in a big fridge, and then you have a low pressure and low oxygen storage. So this is an outline of how to do cryopreservation. You can dip it in a DMSO, dimethyl sulfoxide. Okay, so you have a shoot tip culture and pre-grown on medium with DMSO, and then you can plunge it in medium, and eventually keep it in the fridge after you have put it into the DMSO. Okay, next, the last thing that we're going to look at is somaclonal variation. So somaclonal, when you talk about clone, in your mind, clone should be all the same. However, although all plants regenerated from somatic cells should be clones, there should be something that is going on, something different. And in several observations, it shows that they are actually not clones. Imagine that you have a flask full of plant, same plant, and you thought they should be clones. Well, they should, but however, actually there are variation in these plants. Okay, so somaclonal variation is a great promise in time reduction to produce new varieties or breeding lines which are easily patentable due to novel variation. Because although you thought that they are all of the plants in one flask are clones, but actually sometimes there could be reason that causes them to be different. And this paved the way for you to produce new variety of plants by just growing at the same time. So somaclonal variation is started off with the callus tissue, organogenesis, regenerated plants, and then hardening, and you get the somaclonal variants. And from somaclonal variants, when you detect the difference, you can take the variants and grow into callus tissue, regrow them, so because you know that they have a variation in the traits. Okay, so in this figure, Somaclonal variation, you can see that there are 8 flasks here. They should be the same, but because they are actually a little different, because one of them has just one and small leaf, and some has several branches, and some one of them has lots of branches with lots of leaves. So this shows that although they should be cloned, but they can also be somaclonal variation. So the advantages is it helps in crop improvement, because if let's say you know that one of them has a difference in variation, it could help you to produce new variety without you having to do lots of effort in producing new varieties. And creation of additional genetic variations increase and improve production of secondary metabolites. And secondary metabolites here, like I said before, it is a compound that is different in a plant that you want to overproduce. For example, if you have a ketone, which is different than other plants because it contains mitragynine, and coffee is different than other plants because it contains cathane, and lime is different because it contains limonene, and all sorts of these plants, they are different because they contain their own specific secondary metabolites. So so because of if let's say you want to overproduce the secondary metabolites, you can observe somaclonal variation and you can regrow them so that we can have a version of a plant that that probably can produce extra amount of secondary metabolites. However, there are also disadvantages on somaclonal variations. If let's say you want things to be uniform, you don't want to see any variation in your tissue cultured materials, and because it may lead to undesirable characteristics, and you don't want that to happen. You want everything to be the same. So when one is different, so it will cause undesirable characteristics. And selected variants are random and genetically unstable. And sometimes, if let's say you think they have this kind of trait, but when you reculture them, the genetic is unstable and it will return to something else, or return back to F1 or F2, the previous, the previous clone that you used. It requires extensive and extended field trials, and it's not suitable for agronomic traits like yield, quality, and etc. If let's say you are focusing on traits like yield, you know that clone should produce this much and this much of yield, and it should produce this much of quality, and suddenly when you have somaclonal variation, it destroys your original intention of producing how much yield and quality. And it may develop variants with pleiotropy effects, which are not true. So you want everything to be the same, and you don't want to see differences in them because it is supposed to be clones. All right, and these are the references. Any question you could just ask me, and thank you for listening.